Welcome, everybody. I know this is a big room and a small audience, so we will do our best to entertain you enough so we don't lose any of you during this period, because that would be tragic. This is the session on advances in pluripotency. I'm Jeanne Loring. I'm a professor at the Scripps Research Institute and the director of the Center for Regenerative Medicine there. And most of my work is involved, it used to involve embryonic stem cells, but it is now involving iPS cells. And I have a short introduction, then each one of the speakers will come up. I'll introduce them briefly and talk about their particular expertise and interests. And then we'll have a discussion afterwards. So I hope you stick around for that part, because I think that'll be more interesting than just listening to us. I want to make sure we're on the same page before we start, because there are two kinds of stem cells. And most of the information that you will hear in the press and even at this meeting will be about what are called adult stem cells. The FDA has approved the transplantation of hematopoietic stem cells from one person to another. That also includes cord blood. But these cells, we have to understand, are not able to give rise to all sorts of cell types in the body. They have very limited what we call potential. They have very limited differentiation potential. In contrast, there's another kind of stem cell called a pluripotent stem cell. These cells are able to make every cell type in the body. And so you can imagine that the applications of these two cell types, adult and pluripotent, or whatever you want to call them, are very different. And we are on the cutting edge right now of and my panel members are on the cutting edge of developing therapies based on pluripotent stem cells. So stay tuned, because this is going to be the way for the future. This is the way I like to show the stem cells in general. There's a stem, and then there are leaves. There are things that the cells can do. The adult stem cells, there it is. Adult stem cells from bone marrow can make blood cells. Mesenchymal stem cells that you can get from bone marrow or from adipose tissue. You can actually get it from any tissue in your body. They're essentially connective tissue cells, and they are able to give rise to connective tissue. So they are cells that make the stuff that surrounds cells in the tissues. In contrast, these are pluripotent stem cells. You can see on the left and right lower corners that the adult stem cells are actually branches on the pluripotent stem cell tree. So these cells can give rise to every single cell type in the body, including the precursors for the adult stem cells, which are in use in many applications for blood, for example. But they can also give rise to heart cells, to skin cells, to brain and spinal cord cells, liver, muscle, and in one case, which is, I think, especially interesting, gametes. So if you want to do a replacement and you want the cells to live long-term in somebody's body, then pluripotent stem cells need to be your source. There are two sources of pluripotent stem cells. These are the human variety. The first pluripotent stem cells were made in 1981 from the mouse. They were made from embryos called blastocyst stage. That's before the embryo actually attaches in the uterus. And in 1998, similar cells were made from human embryos using the same strategy. There are cells that will give rise to the entire embryo and to the entire person. And if you can isolate them at the right time and put them in a culture dish, you can expand them and then differentiate them into different cell types, depending on what your needs are. In 2007, Shinya Yamanaka reported that you could make cells that were identical to embryonic stem cells, but you could make them from individuals, from any person, by taking a tiny bit of their skin, growing dermal fibroblasts from that skin, and then converting those fibroblasts into pluripotent stem cells using a method called reprogramming. And of course, Shinya Yamanaka won the Nobel Prize in 2012 for that discovery, which has really changed the face of pluripotent stem cell research. I want to point out that these cells, there's a great deal of evidence that iPS cells and embryonic stem cells, HES cells, are identical in all the functional capacities. There are subtle differences. Those differences are just as great as they are between one individual and another. The genetics of that person, the genetic background, has the largest impact on exactly how those cells behave. Okay, so it's easy to think about what these cells can be used for. We're going to only talk about a couple of these because they can make everything. We can use them to understand human disease, those so-called disease in a dish studies. We can use them for drug development or precision medicine, and that will be the topic of one of our speakers. You can also use them for cell replacement therapies, for example, our project on Parkinson's disease to transplant dopamine neurons into people with Parkinson's disease. And then the last one on the list is one that I won't talk about, but come and see me later if you wish. Because you can make gametes from these cells, and you can make these cells from endangered animals like the northern white rhino, we believe that it'll be possible to rescue species that would become extinct by making gametes from their iPS cells and doing IVF and making embryos. So this is a list of our speakers, and I'll introduce each of them as they come up. The first one is now Terada. He's a professor and director of experimental pathology division, and you had a few other titles that I left out for convenience. But I think importantly, he is the director of the Center for Cellular Reprogramming at the University of Florida College of Medicine at Gainesville, Florida. He's going to talk about the use of pluripotent stem cells for precision medicine. And I'm thinking that he's going to be talking about the genetically diverse iPS cell banks that you can create and how you can use those to hasten and improve the development of drug therapies. Yeah? Oh, here. Oh, great. That's for you.
Oh, great. So thank you so much. Is this the mic or this mic?
That's the mic.
This is the mic.
Don't
oh, no.
into that.
Okay. All right.
Press
Oh, this
the
is for
green
press. Oh,
button.
I see. Okay. So thank you so much for a very nice introduction. So again, my name is Naohiro Terada, and I'm from University of Florida. So I should say welcome to the Sunshine State, although my town is like 5-6 hours north from here. So I've been working with a pluripotent stem cell for over 20 years, but the last 7-8 years, I was working as a core director for iPS cell core to promote the use of iPS cells within the institute or even outside of the institute. So I would like to talk about our effort to how to utilize iPS cells or how to facilitate the use of iPS cells in GWAS-based studies. So as you know, the precision medicine is a way to mitigate the disease, individual disease risks based on the GWAS data or some other big data, or try to find optimize a therapy, again, based on those GWAS data. And can I have a next slide? Oh, should I go to?
Push the green button.
Push the green button, okay.
Yeah.
All right. So pharmacogenomics is one of the very, very important component for precision medicine. And in the old days, we were treating patients with one drug, for example, and then just later, only later, we found the drug was indeed effective in certain patients, but not so much effective in other patients, or some other patients will only develop adverse effects. But now, by utilizing those genome-wide association data and the clinical data combining, we could predict who could have a best benefit from certain drug or who would likely develop a certain adverse effects. So in advance, we could tell who would have a best benefit from certain drugs. That is a kind of effort for pharmacogenomics. So these gene linkage data, genomic linkage data by itself, it's really sufficient for predicting who would have more benefit from certain drugs or who would have a more higher disease susceptibilities. But if we'd like to know the mechanisms underlying, we still need some biological interface to study the molecular mechanisms underlying how particular SNP difference or how particular genetic variation would cause certain differential drug responses or differential disease susceptibilities. So in that sense, induce pluripotent stem cells, which retaining all genomic information from patients. And also they have a potential to differentiate into, theoretically at least, into all the cell types, would have a best resource to utilize for this molecular mechanism studies. So there is actually a pretty good paper published in the last year, or 2016, by Rudy Jenish's group. So there is one particular SNP identified GWAS for the higher risk for Parkinson's disease, and that particular SNP was located in intron 4 over synuclein A genes. But GWAS study itself doesn't tell us in the mechanism how those SNP would cause the difference. But they utilize iPS cells and differentiated in dopaminergic neurons with the proper gene editing. They found one repressor protein actually indeed bind to that particular intron site. When with that SNP alteration or polymorphism, those repressors would not bind that site anymore. So then they have a higher synuclein A gene. So that was really nice in a study for understanding mechanism, how particular SNP would cause a higher risk for diseases. So this a kind of understanding mechanism is really important for us to further develop further like a novel approach or novel therapeutic approach, or knowing optimize more therapeutic approaches. Okay, so for pharmacogenomics research particularly, we've decided to make IPS resources for other pharmacogenomics researchers can utilize more easily and highly accessible and highly practical. So that was kind of the main purpose of what we made a so-called pharmacogenomics IPS cell library in service under the support from NIGMS, and those cells are distributed through the PGRN pharmacogenomics research networks. This is not just US, but international networks. So what we do basically is we made a library with a SNP database, and upon the request of whatever the interest of a SNP, we provide those, basically we provide like a 3 IPS line with their particular SNP of interest with heterozygous genotype. And then I'm gonna talk how the heterozygous SNP iPS cell clone will be very practically useful. But anyway, the goal of this resource is to facilitate the use of iPS cells, again, for SNP validation studies in the pharmacogenomics research community. So this is my last slide, but this is the kind of basic standard procedure. So we actually made a very small number of libraries. It's about 50 from healthy donors. And then we made those from CD34 positive PBMCs, and using Sendai virus. This is from Dr. Nakanishi in Japan. And then the reason we made a 50 is because we feel, again, SNP het clones is very useful for practical researches. And then any of the like a SNP which has a minor allele frequency over 5%, with only 50 clones, we could identify at least like 5 SNP het clones in those libraries. So we could provide like 3 or 4 SNP het clones easily to the investigators, even with these numbers. And then there are 2 methods for the SNP editing, because as you can imagine, this is not a monogenic disorder. So disease linkage with the SNP to some phenotype is very subtle. So we definitely need isogenic iPS cell system. So one system we use is we call the fast track system. So we just do the one allele knockout, and then just showing making heterozygous SNP clones. That is method 1. And this is so easy as a SNP editing using CRISPR-Cas9 system. And then for certain SNPs, such as like protein coding SNPs, or at least for certain intron SNPs, those fast track methods can be effective we found those are very useful. And then the second method is that we really need a seamless SNP editing for the other SNPs. And that's, as you know, some of you who have been working with CRISPR-Cas9 already know, but this is a kind of a time and labor consuming, unfortunately. But we use a kind of a safe harbor side co-transfection system, which at least increase efficiency to at least a 2 to 3-fold higher. So we put the selection cassette in the AAV1S1 site, and then the target site, which we make it seamless or scarlet. So this is a little bit easier, but still, it's not really best approach. But using those methods, we provide either iPS cells or resources, as a resources or like protocols we developed. And in addition to that, we also provide differentiation protocols to individual researchers. And then differentiation protocols, we cannot cover everything, of course. So we provide best protocols we have, or that we just introduce, other investigators to connect with other investigators who has the best protocols for certain lineage. But anyway, this is the kind of our effort to try to make iPS cells more available, accessible to the more, I should not say it's ordinary, but researchers who don't have too much experience with iPS cells. So that is our goal, and then so far it's been pretty successful, and it's getting developing. So I really appreciate NIGMS and also Pharmacogenomics Research Networks for this effort. Thank you for your attention.
Okay, so hold your questions. Our next speaker is a woman who's been in this business longer than I have, which is pretty unusual. Outi Hovatta is a professor at the Karolinska in Stockholm. And she's been working on differentiating cells from embryonic stem cells, pluripotent stem cells of the original ilk, that will be used for central nervous system cell therapies. So I'm looking forward to hearing her insights. Her title is Differentiation of Neurons from Pluripotent Stem Cells for Clinical Trials.
It's a pleasure to be here, and I want to thank for this possibility to tell you what we are doing. As Jeanne already mentioned, we have been differentiating different types of neural cells, mainly from human embryonic stem cells. We do also iPS cells, but in our hands, these ES cells, which we make in clean conditions, good manufacturing practice laboratories, work extremely well. And this is just a cartoon of how we can make, for instance, motor neurons. Geeta Ravindran is a very good senior scientist who has developed systems that we can, without any genetic integration, using just high-quality growth factors in the cultures, make fully functional motor neurons. And the purpose of these motor neurons is to treat amyotrophic lateral sclerosis. We have proved in animal experiments that when we inject these cells to spinal ganglia, they grow axons to muscles and form junctions. And in the near future, we hope that this will be the treatment of ALS, which is a devastating disease, killing the individuals within 4 or 5 years. And then we have been producing high-quality retinal pigment epithelial cells, also from embryonic stem cells, in completely xeno-free and chemically defined media and factors. This is a 6-week procedure, but if we keep high quality, we get large numbers of RPE cells, and the first clinical trials using these cells are ongoing in Stockholm. And these are just experiments with rabbits that have big eyes. And as you see there are nicely integrated RPE cells as shown in this spectral domain optical coherence tomography in rabbit eyes. Rabbits are easy experimental animals, but we can do that also with rodents, which facilitates the work. This is mouse eye, the red is lens. Benjamin Joshi here in Nova Southeastern is conducting this research. And the aim is to make these ES cells tolerable to recipients. We have, for instance, in Stockholm, a bank of more than 300 cell lines, but it is not practical to do all these HLA types, which all the people are trying. And now we can do more tolerable cells. We have been testing, for instance, a method called co-stimulation blockade, which works. And now we are trying to modify these cells to resemble more like placenta that is not rejected by the mother. And we are in that way. And there is plenty to do, of course, but we are quite hopeful that with the existing high-quality ES cell lines, we can treat a large number of patients with good quality tolerable ES cells. I want to thank my collaborators in Karolinska Institute in Stockholm and Nova Southeastern University in Fort Lauderdale. Thank you so much.
So Outi has just come straight from Sweden, and she warned me that she would be jet-lagged and maybe not as quick as she usually is, but I think it was very clear what she's doing, and in fact, we should probably spend some time discussing transplants of retinal pigment cells to treat certain age-related blindness. Those trials are actually going forward in several locations, and I think it could be one of the first successful applications of pluripotent stem cells that is approved by regulatory agencies. Our next speaker, and we're going sort of outward and higher altitude as we go. The next speaker is Liz Schwarzbach, I'm sorry. She's from the New York Stem Cell Foundation Research Institute, which is a very well-known organization that is focused on development of stem cell therapies. She's going to talk about enabling technology that they've developed at the New York Stem Cell Foundation. And one of those technologies is a high-throughput generation of iPS cells and high-throughput generation of differentiated cells. She will also talk about a clinical trial for that very disease that I mentioned earlier, age-related macular degeneration. Liz?
So first, thanks to the organizers of the World Stem Cell Summit, and particularly Bernie Siegel, and thank you, Dr. Loring, for chairing this session. As Jeanne mentioned, I'm Liz Schwarzbach, and I oversee partnerships and collaborations at NICEF. So just by way of background, NICEF was founded in 2005 at a time when NIH funding was not being given for any research that included embryonic stem cells. So to address this issue, the Institute really focused on providing fellowship and investigator awards, and now has a global network of over 140 investigators and fellows, and really looks to convene that group, both with conferences and local programming. In parallel, NICEF has been a safe harbor laboratory and has a research institute with around 45 independent researchers that are really looking to bridge that gap that we've been hearing about around the fundamental research and what's going on in the biopharma industry. So we have disease-focused research in many neuronal disorders that you see here, in diabetes, bone and cartilage, retinal disease, which we'll talk more about shortly, and then rare diseases. I'll spend more time talking about some of the tools and capabilities that NICEF has created to really move this field forward. So first is the automated reprogramming, known as the NICEF Global Stem Cell Array. So this is really a platform to address two main issues in the field, and if you could start the video, that would be helpful. So the first issue is scale. So, as the initial work was done to really optimize protocols, et cetera, these were being done on limited cell lines. So to do this in a more robust way, this global array was developed. On scale, this now can produce, in the order of hundreds of samples per month, so addressing that need too. There's really 6 main components to this platform. First, the automated fibroblast production, generating diverse banks of fibroblasts, and really controlling for growth rate and selecting for low passage to ensure that we have a robust starting point. Then is the automated reprogramming, followed by the automated iPSC purification. So here really looking to have a negative selection against any incomplete reprogrammed cells. Fourth is the automated parallel culture of multiple iPSCs. So there's a second robotic arm that focuses on expanding and freezing down the embryo bodies, and also RNA and DNA for QC quality control. And then automated analysis of those differentiated propensities, so a quantitative assessment of the pluripotency. And then finally, automated differentiation. And this is an area that we've continued to refine over the last couple years. And initially, we started with cardiomyocyte differentiation, and have now moved to several cell types that we can automated have differentiation. So this platform is now part of an end-to-end solution that NICEF is offering for stem cell research. We've created a large repository now from this automated process of thousands of QC lines from consented patients, which I think is a key component here, a valuable resource. And this is all connected to a portal that includes both patient and genomic data, and we've managing this through a program management group. So really, the level of rigor that's put into the cell line banking and the end-to-end cell research, I think, is a unique resource for the community. One specific program that NICEF has been developing is the neurodegeneration program. So within the last year, we've brought in 2 new members to the leadership team. So first, Rick Monsma, who is the SVP of Scientific Operations, and he has a background both at Merck and Roche, was in the neuroscience screening group at Merck. So he's really able to align the timelines of the academic research going on within the Institute with the needs of many of our partners. So I think that's a really valuable resource. And then Melissa Nierenberg, who had been a longtime collaborator with NICEF and have been at multiple New York institutes, has been brought on to oversee the clinical group as a chief medical officer. And one of the unique components to Melissa being on board is that she has built over time this well-annotated, unprecedented collection of human stem cells generated from individuals with various subtypes of Parkinson's disease. So we can talk more about that in the panel, because it's an area of interest. And just on the right, I show a couple of the differentiation protocols that we've kind of been optimizing within our labs, and really working from the earliest stage fundamental research, looking at the mechanisms underlying, with the oligodendrocytes, multiple sclerosis, doing drug discovery work, so starting to initiate target identification and validation studies, looking to be a resource for phenotypic screening, and then, kind of getting back to the goal of NICEF, or the mission of NICEF, to advance therapies to patients. We're really looking to develop replacement therapies for cancer, orthopedic indications, and as Jeanne mentioned, probably furthest along is our work in ophthalmology. We have just brought on board Mike Zanowski, who's in the audience, and is looking to build a GMP facility within our site. So he's right over here, so if you have questions around the GMP progress, and we'll be going forward with a differentiation protocol for RPEs, and looking to address the issues of macular degeneration. So that will be kind of the first area where we build out our clinical capabilities. So that's the end of the presentation, and I just wanted to end with an invitation that, one, we have built a new facility on the west side of Manhattan, so just an open invitation for anyone here to visit our facilities and leverage the convening space that we have built in New York. And then secondly, we do host an annual conference that's really kind of the premier translational research for stem cell conference, and that's in October. So anyone who's interested in supporting or participating in that conference, you can see me afterwards, or the email address is on the screen. So.
Okay, I hope you're thinking about what you wanted to ask these individual speakers, because we'll have discussion afterwards. Our final speaker is Stephen Sullivan. He's with GATE, which is the Global Alliance for iPS Cell Therapies. He is based in the Scottish office in Edinburgh. The title of his talk is Coordination of Activities for Clinical Applications of Pluripotent Stem Cells. So this is the highest sort of level of organization and attempts to organize multiple clinical trials. So one of their major initiatives is to develop standards and methods for sharing information. I think we've heard a lot about that at this conference already, and we've heard how difficult that is to do. So I think it's great that Stephen is working on trying to get us to communicate with each other more easily, more frequently, and without any fear of competition. Stephen?
Thanks, Jeanne, and thanks to Bernie for the invitation. So I thought before going forward, just to, I suppose, re-spark some synapses from the key points in the conference so far, that, you know, that the whole field is getting serious now about changing things for the patient. And the lack of standardization in various stem cell fields is a real problem. And those that don't know history are doomed to repeat it. And smart people don't, make the old mistakes, they make the new ones as they move forward. And really what we need to do is kind of fortify the networking capacity. So at the moment, in order to get an iPS cell therapeutic to the clinic, there are all these individual silos with their individual motivations, their individual languages, and their individual biases. And what we really need to do is to build a collective understanding about what's important in the whole process of moving a cell therapy derived from iPS cells to the clinic. So, in essence, there was one quote yesterday from the Biden Cancer Organization, where he was saying that everyone's on the ball, but no one has anybody to pass to. And that really sums it up very nicely. Another way of actually communicating that whole sentiment is an economics term which is like the tragedy of the commons. And this is, if you imagine a common grazing area and all the farmers with their cattle, they see the common grazing area and they say, great, I'll throw all my cattle out there. And soon enough, the field is totally covered with cattle, depleting the common resources to the detriment of all. So, basically what it's talking about is if you don't do things for the community and you only act in your own self-interest, eventually it'll come back to bite you. And ultimately, we don't have an iPS cell product yet, so collaboration is really the logical way to develop the market and develop therapies. Being competitive before we've even developed a product is actually, it's detrimental to all of us. So where GATE comes in is we're an organization that's funded by 4 organizations that have realized this. So we heard from Susan Solomon yesterday from the New York Stem Cell Foundation. Also in Facilitate, there are speakers from the Cell and Gene Catapult in London, and the Canadian Centre for Commercialization of Regenerative Medicine. And those organizations, along with one in South Korea, have put their money where their thinking is, and they have established GATE. Now, the key thing, what GATE is trying to achieve, is it's trying to build what's called a haplobank of iPS cells, clinical grade iPS cells. And what that means is that you have nodes all around the world that have clinical grade iPS cells that are matched to different patients. And there's a collaboration there where you have, like with, say, bone marrow registries and, stem cell networks and such, that, you can collaborate for the good of the whole. And that's really where GATE is moving towards. So key thing, again, to reiterate is that moving iPS cells to the clinic to do something real for the patient demands that we all actually get together and develop a common language, because, like, for example, an academic scientist will have a very different perspective because they're under, different incentives than, say, a manufacturer. So, one of the great things about Jeanne and Bernie is that they break down these, silos, and that, because ultimately, if they remain, then, for example, an academic scientist, they might be motivated to make iPS cells to a standard that's good enough for them and, like, research grade, but that actually mightn't be of any use to a patient other than as a tool that will indirectly generate information rather than being cells that you could actually put into a patient. So really, the key thing is that we have an awareness of what the whole process is, and talk to, like, we heard from the FDA last night, for example, we should engage regulators as well as letting regulators engage with us, because it's much better for the field if, the field comes to its own consensus over standards in collaboration with the registers, with the regulators, rather than, not engaging the regulators, and then the regulators have to come in at a later date with a set of standards which negates an awful lot of work that's been done in the field. So, GATE, what it's done to date is it's sat down with many of the stakeholders involved in the process of moving iPS cells to cell therapy in the clinic, and it's asked them to come to their own decision on the standards that are important for such a transition. And we've got a set of QC standards by which we will allow iPS cells into our banking network. And now also we're at the stage of thinking about the problems of manufacturing and really getting to the key elements of scale-up, for example. So that, involves a different mindset and involves these different people talking across silos. So, GATE, the key thing with many technologies is the rate-limiting step in all of it is trust between people and understanding between people. So, we're very fortunate to have these organizations to fund GATE in order to try and address that particular human issue, but it's, ultimately can be the rate-limiting, step of the whole process, even though the technology is obviously very complicated. So thank you for letting me speak.
Okay, so we've touched on certain, I think, very important factors here, and I think if we start with some of the biggest ones and maybe go down to some of the smaller ones. This matter of standards, for me, this is reminiscent of the Human Genome Project, which I had a very small part of. The quality of different people's sequence was extremely various, if I should say it that way. And nobody wanted to impose standards on any academic researchers. But in fact, if you wanted to join the club, you had to rise to a certain standard. And I think that sort of thought, which was actually, the idea came from Francis Collins, who is still at the NIH, was that if we all work together, we will all benefit. So I'd like to ask this group first, just to get us started, about what sort of quality standards do you think are the most important to share among different researchers? You can start. Let's start with you.
Should I? Okay.
We'll go this way most of the
Okay, so I'm using iPS cells for more research purpose, so my standard should be very different from a clinical purpose. But we also, because we do the gene editing, definitely we're concerned about some off-target effect of other mutation during reprogramming. So what we do is we definitely do a standard iPS cells differentiation test with kits available from the company. We usually use the stem cell technologies. We confirm the karyotype, and then in certain case we do the gene sequencing for the mostly exome sequence. But we don't do too intensively to test all the genomic sequence because we start from multiple clones from multiple patients and then multiple clones. So by those data, we try not to do too intensively for the research purpose use of iPS cells. Again, this is a more practical purpose for us.
Yeah. Can I just, I'll ask you another question, then we'll keep going that way. And we'll make this last a little longer. So are you concerned with the issue of off-target effects with
Yes,
with CRISPR?
definitely. Yeah, so we use a high-fidelity Cas9, which substantially, very much decrease, but still, there is a chance for off-target effect. So instead of doing a sequence, we prefer to use multiple clones. But still, by those, there is a chance certain guided RNA would cause the same off-target effect on that different clones. So we cannot really escape from all the issue. But doing all the sequences is not practical for us to confirm. So that's why we use practically multiple clones as a kind of solution for our research purpose use of iPS cells. Yeah.
Right. And the standards may be different for research grade than for clinical grade, because the issues are different and the outcomes are different.
Right.
Okay. Outi.
Yes, we are now working with these embryonic stem cell lines, and we have very high quality standards if we think that the line will be used in clinical treatment. So how they are made in good manufacturing practice laboratory is very strict, and we follow all these international standards. And we test them very carefully so that we know that they don't form tumors or something in experimental animals, and that they are functional. Functionality is one of the most important factors. It doesn't matter if we have hundreds of lines if none of them really functions in practice. And that's why we do a lot of animal testing. Mm-hmm. Before we go to any treatment, we of course sequence every line. We have a full sequence from all the lines we have made.
Yeah, that's something I want to bring up separately in a few minutes about the genetic testing of these cells. Liz?
Yeah. So for all of the lines that we make, whether they're research use or otherwise other applications, we do have a certificate of analysis for every line, and if it doesn't pass that QC, we won't distribute it or put it in our repository or use it within our labs. So that certificate of analysis would include looking at the established markers of pluripotency, the karyotype, depending on what line it is, or if it's a differentiated line, or if it's from a patient, what that disease is. We'll have different types of QC. So for instance, we may look at genetic markers of disease to make sure, in fact, the patient didn't just have the phenotype, but has the genetic makeup for that disease. So we will look at every line that we produce regardless of use.
Yeah. So just a follow-up question, because one of the things that I think most people wouldn't realize is that there is a fairly high probability that the frozen cell line that you obtained from a vendor or even from a colleague will not be what you thought it was. Yeah. And so I think one of the things that we've learned from experience is that if we get something from somebody else, we always, before we launch into a huge project, we make sure that that cell line is what we think it is.
Yeah, so now, one of the things that we're doing more and more is as we're working with collaborators, will typically request more samples than, particularly for the automated system, we'll ask for more samples than what they're looking for at the end, expecting that a few of the samples may not be what we're expecting to receive, so that, if there are ones where that is the case, we're still able to deliver at the end the number requested. Initially, even talking about success rates and kind of making sure that you do come out at the end with what you're expecting. Initially, just on the automated fibroblast production, we had about a 90% success rate. We were able to continue to optimize that, so now that is almost if the sample is what we expect it to be, is almost 100%. Maybe it's just slightly under, but kind of continuing to refine the protocols to get to an increased success rate. But that is something that we look closely at.
Great, thank you.
I think it's important to reiterate that it's a very different world between developing a research tool and developing a set of cells that you're going to inject into a patient. If you're using a particular test to test some facet of your cells in the lab, it might cost $200 or $300 to use the kit to test for something. But that kit mightn't work 100% of the time. And if you're going to manufacture cells that you're going to put into a patient, you have to be damn sure that the kit is going to work 100% of the time. So there's a huge difference in cost in generating an iPS cell line as a research tool and as a cell therapeutic or a derivative. So it costs around, I think, $1 million to develop one clinical grade iPS cell line. So it's not a frivolous undertaking, but unfortunately, the key thing is that if everybody does their own thing to a degree, then there's a huge amount of waste. And if we don't get on the same page at the beginning of this process, a lot of people's investments will go into something that ultimately doesn't do anything for the patient. So it's just really important that we get on, we understand the same things from language, for example. Like if you want to get really detailed into it, we cannot have a GMP iPS cell line until there's a product in the market, because you need a product in the market to get that accreditation. And people are talking about clinical grade and GMP iPS cell lines, but they mean different things by that terminology. And those kind of slight misunderstandings or incongruities of understanding of language have a huge difference later on in the process. So we really, in order to make sure as much of the money as possible goes into something of benefit to the patient, we need to all develop a common understanding of the whole process and learn, have the defined language that means the same thing to all parties.
Yeah, good. So there are just a couple of things I want to add, and then I'd like to ask you questions from the audience. One of the things that we are really considering, especially Stephen, are the differences in regulation among different parts of the world. And it is difficult enough to understand what the FDA requires. But if you're also looking at the European Union, if you're looking at Australia, if you're looking at Japan, there isn't a homogeneous approach. There are things you can do in one country with approval, and you can't do that in other countries with approval. So it's the only way I learn these things is by talking to people who are trying to get approved in other countries, and they tell me what sort of things they've run into. I think it would be nice to have that knowledge a little bit more easily available. I know those are some of the greatest secrets. Regulatory agencies, especially the FDA, I think, don't really like to tell you what to do. They want you to ask to tell them what you want to do, and then they say whether or not you should be doing it that way. And that's a very inefficient way of coming up with the right strategy, but it is what we have, so this is what we're doing. The other thing I wanted to mention is really probably down in the weeds, but there have been some issues with even cells, iPS cells have been used in research because people didn't anticipate certain characteristics of iPS cells. Some of those are that they acquire mutations as they're expanded in culture. Some of these are very large. Some of these are oncogenes, which are things that we should be concerned about if we think the cells will become cancerous. We don't know for sure, but it is something we need to consider. And there's also the matter of mosaicism in disease. So Down syndrome, for example, is trisomy 21, but not all the cells in a Down syndrome patient are trisomy 21. And it is perfectly possible and has been done that you get diploid cells from a Down syndrome person if you make iPS cells from their skin. There's also the epigenetic issues that have haunted some people who are studying X-linked diseases, because they didn't realize that if you have a female patient who has 2 X chromosomes, they start out, their somatic cells have only 1 active X chromosome. But after you culture them for a while as iPS cells, there's a fairly high probability that they will activate that 2nd X chromosome. Now, if you're looking at a recessive disease, that is, that somebody actually has in a woman, and you realize that when you start out, you have mutant cells, and then after you've cultured them for a fair length of time, you suddenly have wild-type cells. That can be quite a shock and a disappointment to researchers. So there are certain quality control measures, or maybe anticipation of problems that researchers have to do, as well as a whole nother set of things on top of that that people who are using cells for clinical research need to consider. So with that, people may want to comment on what I just said. So.
Yeah, just one other kind of point that is interesting to, like, from a basic research perspective, if you're generating iPS cell lines, you'll want to do full genome sequencing on them, because, as a basic researcher, you get as much information as possible. But the manufacturing/engineering perspective is quite different, actually. What you want is enough information that you know where you are, but you don't have a surplus of information that you don't know what to do with. Because as a manufacturer, it's about mitigating a variance and controlling things and making things robust. And if you have a huge amount of information that you don't know what it means, that's going to slow down the whole process, and it's going to take a lot longer to get something into the clinic. Also, you'll have a very hard time with regulators when they're asking you about questions about data that you don't know how to interpret. So really, we have to be careful that what seems to be the best way for us might actually not be in the context of the full process. And we have to, really think about the full process in order to accelerate things into the clinic as quickly as possible.
So as a counter argument, those of us who have a genomics background or sort of steeped in genomics don't have a really hard time with whole genome sequencing, and so we are planning to include it as part of our manufacturing process. But the project that we have planned is an autologous therapy. That means that we don't need to make 1 billion cells. And so doing whole genome sequencing, I think, is going to be, even though we do it on more than, we do it on every single person's cell lines, we do it at the beginning of the fibroblast, then we do it when we make the neurons. And we will be doing it for all the preparations that we make for GMP. So once you have that information base on individual cells, it's not, if you have good bioinformatics experience or expertise, then it isn't that challenging. There is a challenge with educating the FDA about the meaning of those data, because I'm sure most of you have heard about the random mutation occurring in some iPS cell line from some patient, and that will shut down a trial. That's what happened in Japan. In that case, the mutation that arose in that patient, or was discovered in that patient, was not anything to be concerned with. And I like to point out that all of us are so variant that you could shut down any of us because we're not the same as the other one of the standard. So we don't really have a standard. What we have is a standard of how much variability and what is normal and what is not normal, if that makes any sense.
Yeah, and I think the one other kind of component to think about, and then you talked about speaking to some of the challenges as well. You know, when you're talking about QCing a line for research and kind of, you know, there you're focused on the product. And when you're thinking about QCing or, you know, kind of creating a GMP line for clinical use, it's much more about the process than the product necessarily. So really, you know, kind of having a standard around every step of the process. And one of the difficulties with that is when you're going into a pre-IND meeting, that leads to a pre-IND meeting, that's before the IND, that's before you reach the clinic, you're making the decisions around the process, you know, in a landscape that's moving so quickly, much before that process is ever going to be used. So you're making decisions around what differentiation protocol you're using, potentially 2 years before it's in the clinic, and there may be a much better protocol at that time. So I think also, you know, working with the FDA around how are those adaptations going to be looked at for a comparison to adapt into a trial, if that is going to happen. And as you do validate every reagent that you're using, every component of that process, how adaptable we're going to be given the time frame of when it's actually going to be used in patients. So I think, you know, that's one other component to the standards and to the process.
That's an excellent point.
I mean, just
Yeah.
one other point was that, you know, probably regulators in one jurisdiction will get certain things right, and other regulators in another jurisdiction will get other things right.
Yeah.
So probably, you know, what's going to happen is that different stages of the process will probably be better done in certain geographical locations. Like when you see like standard traditional drug development, it's usually done in several different countries, clinical trials in several different countries. And it's probably going to be the same with cell therapeutics as well, which, you know, it can be a difficult thing in terms of, you know, if your local agency wants everything done in your local geographic location. But really, you know, our end goal is to cure patients and to treat patients effectively. So we have to, you know, ultimately go where the progress can be expedited most efficiently.
Any other comments on that
I
subject?
think the one other thing that you brought up is to, you know, using an autologous therapy, and even those decisions have implications around then what your indication is going to be given the timing. So just, I mean, I think the difference between kind of bringing a cellular therapy, particularly an iPSC, to a patient and considering those variables is much different than, you know, having a target product profile for a small molecule where your indication is defined and then you can adapt the program to that indication, where here, you know, if you're going to do an autologous therapy, you know, the type of macular degeneration that you actually can treat changes given the time frame of when that treatment will be delivered. So I think, you know, kind of the complexities are much different in how they relate to one
There
another.
are many moving targets, all at the
yeah.
same time, yeah. But we can handle it, right?
Yeah.
As a team.
And I
Yes, as a
think-
team, we
yeah.
can handle it.
So maybe one of the areas that we're, you know, since I was talking to the challenges, one of the areas where I think we are making advances as well is, you know, moving from just the skin biopsy to using any sample from a patient. So now reprogramming from blood, you know, we initially were using Sinai virus, but also now looking to use mRNA to do the reprogramming there. Even looking at, you know, whether it's doing collection of urine, saliva, et cetera. So expanding to, you know, the sources for iPSCs, I think, is another area where we're looking to kind of make.
Yeah, no,
some advances.
that's an excellent point, because you may not have urine. We've made iPS cells from urine, and we did it just
Yeah.
on principle.
Yeah, exactly.
Right, and so we know that can be done, at least for the people we've done it with.
Yeah.
I think that one thing that the rest of the audience may not be aware of is the methods that we make to the iPS cells. We want to use methods that don't affect the genome of the cells and known. So we don't want to deliver any viruses that integrate into the genome. I mean, that's just sort of a principle. It just sounds like a really stupid thing to do. It may not make a difference, but if you have an option of not integrating the DNA, then we think it's a good thing. And so far, there are three really methods, I think, that I know of. I guess there's one that just got published, but I haven't read the paper yet. That you can use Sendai virus, which doesn't integrate but has a very high efficiency. You can use messenger RNA, which codes for the reprogramming factors. Both those cases, the what you introduce has to be, you have to explain to the regulatory agency how that was produced, because even though we're not integrating anything into the DNA, we might be bringing along something that arose during the manufacturing process of making that vector to reprogram the cells. I mean, I may be getting a little bit too subtle here, but I think we all know what we're talking about. There are other methods that are also they haven't been as well tested as those. And really, it's just a matter of making sure that you're looking to the future and that those cells will be usable by clinical as well as research purposes. Okay, so does anybody have a question before we keep talking to each other? Yeah, I think you just need to yell, because I don't know if we have any microphones. Just speak loudly.
So you mentioned at the beginning that there's some subtle differences between iPSCs. Does the panel think it's feasible, I guess for some indications at least, that a iPSC-derived product could be developed where the source fibroblast or the source cells used to make the iPSCs didn't have to come from that specific patient?
He's loud enough, that's fine. Sorry, would you repeat the last part, the question part?
So could
okay.
the cells be non-autologous?
Oh, yeah. Yeah, and I think most of the therapies being developed now with iPS cells are, in fact, allogeneic. There's a couple of ways that you can, right now, if you are permanently going to place pluripotent stem cell derived cells into a patient, and you want them to last for the rest of their lives, you have to immunosuppress those people, even if you're putting cells into the brain, because they will be rejected, and that's obviously not what we want. There are efforts being made to soften that rejection by using cell lines that are matched for HLA type, for MHC. And that is more of a challenge in Europe than it is in Japan, because they have a less diverse population. There is also an effort underway to remove the antigens that cause rejection from the cells. That's relatively recent and hasn't been very well tested. And we just need to know that we understand what's going to happen when we put the cells in, and know that we can handle it for the best health or best care of the patient. So you want to comment?
Yeah, no, sorry, go on. Is that you?
Yes, these new methods, how to make them non-immunogenic are underway. It's not necessary to have so many lines, because it's not just removing HLA. There are other ways. And it's not so, in the future, it's not so dangerous as people think today. That's a fast-moving field, so we are going to get there.
Yeah, I think it's a perfectly reasonable alternative strategy.
You just whatever works over time will be the way forward.
Our rationale was that we were as certain as we could be that an autologous therapy would work. And so even though there's a lot more work up front in order to develop that, we thought that the probability of actually treating people and having them have a good outcome was worth it, because it would be much higher. Anything else you want to?
So yeah, I mean, from a proof of principle point of view, autologous is the way to go. But then when you want to get a product off the shelf that you can give to many patients, you'll either have to go the allogenic matching route or the universal donor route.
Yeah, we were not thinking of shelves when we started. So.
Yeah.
Universal donors are not needed because there are better ways to overcome it.
I mean, I think the key thing that I'm trying to get across here today is that there's this wealth of information in cell manufacturing that isn't intuitive to people who have not done it themselves, and things that maybe seem illogical to people in the stem cell silo actually make a huge amount of sense to the people in the manufacturing silo. And if we take those two things down and share information, we'll get the best efficiency between the two.
Yeah, and I think this particular meeting is very good if you want to learn what the other points of view are, because we're partnering with Facilitate, And they don't have a lot of experience with the kind of stem cells that we're using, but they have a great deal of experience with CAR T therapy, for example, and expansion of other cell types. And we should be able to take advantage of what they have learned and what all the expense that they have, everything they've invested in making sure that they get it right the first time.
The discussion yesterday about chemotherapy
Yeah.
versus immunotherapy
That was pretty amazing.
also shows the random nature in which certain forms of research are favored over others. And it's, you know, it's usually kind of short-term ease. And ultimately, I think what we all here at this panel would say is that, like, let's try every avenue we can. And then, you know, whatever works, works.
Yeah, we all have the common goal that we want to do the best possible treatment for patients, because that's what's going to make us feel like we've accomplished something in our lives eventually.
There's a question.
So this is a question to Professor Loring. So you did mention that you are regularly incorporating a whole genome sequencing step
Yes.
to characterize your lines.
Yes.
Now, if we go by what Professor Terada was saying, that he's using CRISPR-Cas9 to modify the cells, in that situation, if you do whole genome sequencing, are you sure you are getting enough depth coverage for looking at off-target? Because it's not a very predominant effect.
Yeah, I understand what you're asking, because the CRISPR-Cas9 has surprised a lot of people in off-target effects. Some genes, there's more off-target, others there aren't. There are sometimes absolute surprise in which the gene you're targeting isn't targeted at all, but it's some other part of the genome is targeted. And you won't know that unless you look at the whole genome. Certainly, we're being very cautious in our approach, so we're not doing any gene corrections in this first run. But obviously, that's something long-term that we were going to want to do. And luckily, the CRISPR-Cas9 seems to be getting better all the time, better fidelity all the time. But I wouldn't trust it. I mean, I would take the most high fidelity approach, and then I would still do sequencing to find out if anything happened to the cells.
Because there was this mention about educating the FDA about the science.
Yeah.
And
No,
I think
exactly.
It's very critical, because when you are trying to go for a huge breadth, you may not get the depth, and one
Right.
needs to be careful about that.
Yeah, and the FDA now understands transgenes because of CAR T therapy, but they don't yet understand CRISPR modifications and how different that is. Yeah, you're absolutely right. And it's our responsibility to educate the FDA, and we're happy to do that. We really are happy to do that. Any other comments on that particular question? No. Any other question? Yes.
I think I know your answer to this, but you mentioned jobs for the Sinai virus for messenger RNA programming. And if you were a new lab starting off, you know, what would you recommend, and how do they compare in terms of variability across lines? What's
Yeah,
the less variability?
Basic perspective.
Yeah.
Yeah, okay.
So I would use Sinai virus because it's one of the, I mean, it's not integrating, and there's a lot of data on it. And there was a paper last year that did whole genome sequencing comparing different methods of reprogramming. That came from my lab, of course, so I think that it was really solid work. But we found that, in fact, that reprogramming cells did not consistently introduce any mutations, and that anything you found in those cells probably arose from this cell population, because we got to keep in mind that we're starting, these are clones of single fibroblasts or single blood cells. You guys actually combine several different ones. And I think you have, that solves some problems, but maybe creating others.
Yes, we'll do pool populations.
Yeah. So we're dealing with a clone, and we're also dealing with the time and culture, so things will change. And the problem is we can't detect, I mean, if you see some really serious mutation in your cells after you've reprogrammed them, you may not detect it in the fibroblasts, but it might have been there. It might have been the 1 in a million fibroblasts that you happened to pick when you reprogram the cells. But one of the things I think it's important to note if you're just starting out is that you don't need all that many colonies, so efficiency, if it's good enough, it's good enough. You don't want to look at 100 colonies. We routinely look at 3. And if there's something wrong with all 3 of them, we'd rather reprogram than have another 97 to look at, because I don't think it makes any difference. And if we won't have to do as much work. Does that sound okay?
Yes, that's great. No, I was thinking you were going to say messenger RNA, but I.
Well, mRNA is good too, but we were trying to, at the time we first tried the mRNA, it was still it wasn't a commercial product, it was still in the hands of the developers. And it wasn't as efficient, and you have to apply it over and over again. So we just opted for what made the most sense for us at the time. And so one of the things I think we're moving away from is the use of just naked DNA. And the problem with that is that it works, but there's a certain amount of integration that you can't be sure of unless, again, you go back and sequence the cells. So this is the safest, most efficient way, and we like it for that reason.
That's great.
You guys do reprogramming. What do you think?
Well, I think Sendai is the easiest.
Yeah.
So practically, this Sendai is really easy. So if you purchase Sendai virus available from a company or from collaborators, you can just get the Sendai virus itself. Then all you have to do is just mix virus with the culture cell. So I think definitely that's the easiest. And then it should be one of the safest method.
Yeah, we do both. I mentioned before that on the skin samples, we moved to doing more mRNA reprogramming. On blood, we're still using Sendai mostly.
And the Sendai virus there's actually a GMP version of Sendai virus. We want to start from scratch now. When we reprogrammed our cells, that wasn't available, so we have to do a lot more testing, essentially the same testing people do in GMP. But we have investigated that, and it's very unlikely that in the preparation of that virus anything got in that we don't want in our iPS cells.
That's great to hear.
I mean, it's important to register too that if you do your process 99% to GMP grade, your cell at the end of the day isn't going to be GMP. It's either all or nothing. It's a binary situation.
You still have to do the testing, and it costs a lot of money.
Yeah.
Yes.
Hi, just a quick question. So talk about IPS lines in GMP or clinical grade, and I'm just curious, I know of 1 or 2 trials that are ongoing that are using cell lines derived from IPS. I was wondering if the panel is aware of, or how many different human trials they're aware of that are going on with such cells.
Stephen, you probably have the global perspective here.
Not too many as yet. I mean, again, what Susan Sullivan was talking about yesterday was kind of the main kind of disease areas. So one of my slides was myopathy is one of the key things we need to treat to move iPS cells to the clinic. And I wasn't just talking about the disease there. I was also talking about the blinkers that we have in our different functions as well. But I think, early stage clinical, I thought it was really important there yesterday as well, where they were talking about clinical trials not being advertised as a treatment, that they are really research, and to modulate, expectation appropriately to patients there. Yeah. I think the vast majority of clinical trials unfortunately yield negative results, but that's the way we get things into the clinic that are safe and efficacious. So not many is the answer, not many.
Yeah. Not many?
Are there any in the U.S.? iPSC? I mean, I know there's embryonic stem cell.
Apparently there are, but I don't know very much about them.
Mm.
I mean, I think I don't think there's anything that has gone to the IND stage, but I could be wrong.
Yeah, that's what
But there are some at pre-IND stage.
There's many, pre-IND.
Yeah.
So not actually in the clinic yet in the U.S.?
No.
Yeah, because
No,
I was aware of
but-
the allogeneic
Right
trial.
on the verge, and there are iPS cell therapies being used in Japan. The macular degeneration study they're doing there is an iPS cell line, not an ES cell line.
Right, and there's the one in the UK, which is the MSC line for GVHD. I think that's Sinai, but I'm not aware of any others. I was just curious.
Yeah. So remember that MSCs are different from pluripotent stem cells. So we are not, but they're made from iPS cells. That's the company in Australia that's doing that. That's right. So you can make lots of things from iPS cells, and one of the things you can make are mesenchymal stem cells, which I like because that means you have more control over it. It's not a batch-to-batch variation. I guess time will tell whether it actually is superior to what people are doing now, which is manufacturing the cells each time for each patient.
I think the good trial watch kind of, if you get on the newsletters of CCRM and ICEF and Catapult, you'll learn very quickly about the real trials that are coming through. So those three organizations.
And I don't know if anybody, one person I think so far has told you not to trust clinicaltrials.gov. We should reiterate that. Those are not necessarily clinical trials. There's no link to the FDA. Anybody can post a trial there. It could be an observational trial. And unfortunately, a lot of the clinics that are offering therapies for by paid patients are using clinicaltrials.gov as a marketing tool to give them legitimacy that unfortunately it doesn't.
So one of the, the main ways of looking and distinguishing between a real therapy, stem cell treatment and a scam would be the kind of the paperwork surrounding it, like that there's a strong scientific and medical kind of paper trail that details all the various things that we've been talking about here today from terms of a safety and efficacy point of view. And also you shouldn't be charged up front for treatment. And if there's no aftercare, that's also a real sign that you're going and getting scammed if the doctor isn't following up on your condition afterwards.
So just a follow-up question for the conversation going on right now. Kind of within the field, these issues with the scam clinics and clinicaltrials.gov can be well known via dissemination through conferences like this. But what about all of the patients out there that are completely removed from the field?
Yeah.
What kind of, if any, measures are being taken to try and disseminate this knowledge to them?
Yeah. Blogs.
Yeah.
Sir?
Yeah.
California Severe Degenerative Medicine tries very hard to educate people. There is a law in California now that's going to require those clinics to say that they're not FDA approved in large enough print so people can see that, because they may not necessarily say they are, but they imply it, and then having a sign somewhere presumably will scare off some patients. I don't know how effective it will be.
Yeah.
But the problem is with any of these clinics, and this is something I do myself, is that I will not name a single clinic because I will get a letter in the mail immediately suing me for libel.
Defamation.
And so I only talk in generalities.
So ISSCR has a really good patient
Yeah,
handbook.
they do.
So if you have anybody who is even considering this and they want to figure out whether it's a legitimate treatment or a scam, that handbook is really, really good. It's got a list of questions they can ask their clinician and it'll differentiate it really quickly.
Yeah, I think
What you won't find is a list anywhere that anybody publishes that says these clinics are scams.
Yeah.
And the reason for that is litigation.
Yes, so I think one of the things that Dr. Marks talked about last night is the FDA now does have a regenerative medicine guidance and does have a mechanism for stem cell therapies to be approved. So now that that guidance is in place, they'll start looking at all these clinics that have existed. And I think they're going to start with ones where there's high risk, so that there's actually damage that has been done to start giving warning letters. But it's going to be a 3-year time period for them to actually go and start to, really be able to provide the oversight to these clinics and actually, shut down some of the clinics that are operating that don't have an approval. So now that step will be taken, but it will take 3 years' times, and they will start with ones that have the high risk. So if it's, ones where it's just not efficacious, that will be a longer term versus where there's a safety issue. Those will be the ones that are addressed in the near term.
So there are, according to the recent surveys, there are around 600 of those in the United States that aren't approved by the FDA. And I'm wondering in 3 years, we could place a bet. Will
Yeah.
there be more than that or fewer than that because of the FDA regulations?
Well, I think it's probably going to cycle, so some will close, but others will open. And
Two
this-
will open for everyone.
Yeah, ultimately, and then, you get these court cases that will incentivize other unscrupulous individuals kind of saying there's money to be made here and go in there. So really, I think the key thing is that we just have to keep repeating the same things over and over again and trying to empower patients with knowledge for themselves to ask the questions.
These clinics are a franchise. A lot of them are are essentially, somebody has a weekend, they have weekend training somewhere, nice hotel, maybe Hawaii or something, or ski season. And then they open the clinic based on that very little knowledge. And the physicians that are involved are not stem cell scientists. I don't think any of them are. They're usually involved in, they could be any kind of physician. I guess I don't want to go too much farther.
No, go on.
They promise a great deal, and they don't deliver. I'm not saying that all of these treatments are not going to be useful eventually, but I think the FDA made it very clear, the guy from FDA said that mesenchymal stem cells shouldn't be in the blood. They're not normally in the blood. And so they are going to be regarded as drugs because they're not being put into a place that they normally are. I think it's a very simple way of looking at it. That's the way the FDA looks at it, and I think that sort of draws a line in the sand for what sorts of therapies you can imagine would be useful and ones that are probably not going to be helpful at all.
And then one other, on the question around the clinicaltrials.gov, the FDA also, actually I think the NIH did say that they have been given funds as part of the 21st Century Cure Act to begin to cull that database. Again, I think that's probably optimistic, Jeanne, I don't know. But they, apparently they have applied resource to actually only having registered trials on that site, but the timing and how effective that's going to be is questionable, but they have committed resource to that.
It's unfortunate, but in the modern age, you know, the World Wide Web and such, it's not just a stem cell related issue. Like, I'm sure if you Google CRISPR cure, you'll probably find lots more a new wave of unscurrilous, you know, websites that'll be trying to take your money.
DIY CRISPR.
Right.
Do it yourself.
Yeah, and I mean, this is one of the worst things about being in this field is the kind of emails you get in every day of the week, where it's, you know, I've got condition X, what can be done for it, and responding that, you know, it's in trials, it's not ready to go prime time yet, and you know, I think we all look forward to the day where it's all past prime time and everyone's just forgotten that it was never curable in the first place.
Yeah. I get several of those emails every week. You probably get more.
Well, I get a lot in Ireland because we don't have a strong science education and medical background. So, you know, you got your NOVAs and all the other brilliant programs.
Yeah. I usually get messages from people have already done their web research.
Yeah.
And they just want me to tell them whether this thing they plan to do is legitimate or not.
Yeah.
Is likely to help.
Yeah.
Excuse me, I have a comment.
Yes.
I think one of the variables as far as slowing down the growth of the bogus clinics, as we call them, would be getting a legitimate treatment through the FDA as quickly as possible. And anything that all of us can do to help facilitate that, I think, would be important. I think patient advocates and different foundations and all of us should be advocates for real stem cell treatments. And on our website, we do have questions that people should ask at the foundation I represent. And just be educated and ask the right questions is the best defense you have.
Why don't you say the website again?
Website, I know I left it out. It's summitforstemcell.org.
Hi. Is this working? This may be a question for Stephen, but it kind of relates to the last topic. I mean, I think we all know about the kind of the problem in the Wild West of stem cell clinics. And then, Stephen, you mentioned, let's say, the problem of various regulatory bodies around the world with different standards. And I sort of heard the inference of the problem with varying results in real science, if you will. Has anyone given any thought, and maybe GATE is the place for this thought, but to common testing standards and common testing frameworks that could perhaps unify collaboration around rigorous scientific measurements?
So, like, you know, in essence, that's what GATE is trying to do. It's trying to get the community to coalesce around global standards for, you know, for quality in iPS cell therapeutics. And ultimately, I think some of, our stakeholders are all subject to their own particular motivations. Like, for example, I was having a discussion, with an associate who wants to fund GATE yesterday, and they were saying that they're having real problems convincing their boss because we were helping everybody potentially rather than just them. And ultimately, what you want is to distinguish between, we all want just the market to be there, and then, there'll be a lot more funding for people like Jeanne, and, the manufacturers will be busy all the time and the patients will be happy. But really you need, big picture thinking there to realize that it's in everybody's interest to work together. But many people's, KPIs are, I differentiate myself from others by looking better than them. So, that's really the problem. And I think that, the patient end of things can really help in that regard, because, competition too early in the process doesn't accelerate things, it deaccelerates things. And as, Arthur Kaplan was talking about the MSc field yesterday, we've got to learn the histories from the other stem cell fields and not repeat them in these new stem cell fields.
I have a specific example of how that worked for us. Although we had no idea exactly what we were doing, we developed a genome expression or gene expression assay for pluripotent cells back when the embryonic stem cells and iPS cells were just starting to be produced. And we thought that the assay that everybody was using, which was to inject the cells into a mouse and see whether they made a tumor called a teratoma, and what the composition of that tumor was a waste of time if there were an alternative. So we invited a lot of people to send us their samples, and we developed a gene expression assay for pluripotency, which we call Pluritest. We have, in fact, licensed that just this year to Thermo Fisher. They're going to be offering it directly to anybody who wants to analyze a sample. We're doing a similar thing with our dopamine neuron development. We're calling it Neurotest. And we've invited, again, this is just being charming to people and promising to publish with them. It's nothing official. We've gathered from all the major groups that are planning Parkinson's disease therapy. We've asked them to send us cells, and we've analyzed them. And that's the core of our neurotest, which we will also offer, as we did Pluritest, free for people to use if they want to compare their cells with everybody else's.
Perfect. So you've basically developed sort of micro-reference platforms.
That's right.
That do, in fact, become a basis for competition.
Yes.
Because you've got the definition of how to measure competition.
That's right. We offered it for free, which I think gave us a great advantage.
Sure.
Among researchers, anyway.
Right.
Yeah.
I mean,
So the real question, so could you take that example, that model, and scale it
Oh, yeah, absolutely.
to
Yeah.
more significant, comprehensive depth and breadth across the industry, which would then in turn potentially help all these different regulators by establishing fact patterns?
Absolutely.
Well, again, I think the key thing is that you can try and force that, but ultimately people will, like for example, we'd have no authority in people over the regulators in Finland, for example, or the regulators in Ireland. And ultimately, the model that GATE is going for is just highlighting the scientific quality advantages of working together, and then asking all the people that come to the GATE community to contribute to formulating a common standard for QC that they will voluntarily adhere to, because they realize it's in their interest, and over time their research and their translation efforts will be a lot more fundable. And it'll drive down cost of goods and other elements like that. But what we're doing later on this year, for example, is we're doing a quality round where we're sending out kind of cellular material to our associates, and they'll be kind of testing those samples according to the quality control criteria that we've agreed. so that everybody understands that they're on the same page, like that, you know, they get the same readings from the same tests of the same cells. And that's the way things are conventionally done to standardize things with blood products, for example.
And then I would just say, you know, at NICEF, that's one of the reasons that we focus so much on convening. So we require any of our investigators or fellows that have received funding, so, you know, the over 100 investigators and fellows, to attend our annual retreat, where, you know, I think the point there is to share protocols, to, you know, share understanding of what, you know, the gold standard is for each, you know, differentiation or reprogramming or whatever the factor that you may be talking about, the neuro, you know, test or et cetera. So I think that's why that is such a focus. And I think, you know, again, we're kind of a victim of success in how fast this field is moving. So you think about, you know, the Nobel Prize for DNA was given to Watson and Crick in the 1950s, and the Human Genome Project was a half a century later, you know, in the 2000s. And here, you know, iPSC technology was, you know, really kind of first published in 2007. The Nobel Prize was in 2012, and here we are 5 years later discussing entering the clinic. So I think as you condense those timeframes, kind of standardizing procedures, et cetera, becomes more and more difficult. And so I do think it takes a lot more initiative from, you know, PIs like what Jeanne's talking about to find, you know, those people who are willing to collaborate in moving the field forward. And I think that's what's going to kind of get us to the level of rigor that's needed to have a clinical product.
Yeah.
Go for it.
Yes. Oh, sorry. Somebody else has the microphone, sorry.
And then
We'll
hi.
get to you in a second.
This is a follow-up to the last comment, the last question regarding established frameworks to start testing methodologies, different sites. I would say that this is something that is being started, and an example of that is the Japanese consortium firm Concept, who is doing multi-site studies of in vitro and in vivo methodologies to test iPS cells. I'm with the Health and Environmental Science Institute. We have a committee on cell therapy safety, and we're working with them and collaborating with them to help them disseminate the data that they are generating. And there is a poster later, if you want to stop by at 5:30, look for the HESI poster and CT tracks. I'll be there and be happy to talk more about that. And they are planning to release some of their data later this year, so be on the lookout.
Great.
That's good to hear. Thanks.
So I come from a culture that's very, very skeptical of science for some good reasons and some not so good reasons. So my question is, I know we can't call out and say this is not a good place because we would get sued, but is there some regulatory office or something that we can contact and report that's not like Better
Yes.
Bureau, or what is that? So that would be really helpful
I
for
think
us
you've
as
probably heard this. The FDA welcomes your reports.
So-
Yeah.
Oh, yeah.
And in
absolutely.
fact, if you ever want to get any movement from the FDA on a particularly heinous clinic, you need to be the people who have gone to that clinic, heard about the clinic, report it to the FDA, you'll get their attention. And who knows what will happen after that, but presumably they'll go on a list and maybe be near the top.
This is one of the things, when they go to clinics like that, patients are obviously and understandably preoccupied with their health. So if they get ripped off, they're usually embarrassed and also more concerned with their immediate health matters than to report the thing. This is how it sustains itself over time, because it's hard to find, and ultimately, once you do find it, you know, it'll move somewhere else.
I know we've had not so many problems of things being done here in the States, but we've had people leaving the States and going to other places for things like bone marrow and other types of treatments. So,
doesn't have jurisdiction there.
right,
I think the
right.
key thing to kind of persuade a cynical mindset in that regard is to say, well, if they could cure disease X in jurisdiction Y, why isn't it everywhere around the world? Because everybody would be, all the inventors would be making a lot more money, a lot more patients would be being cured. And if it's just in one geographical location, that's very kind of suspect, especially if it's a jurisdiction that's not strongly associated with strong medical regulation and oversight.
So I think there are two sources, obviously, of information that's maybe more informal. On our coast, there's California Institute for Regenerative Medicine, who I know there's somebody in the room right here who fields questions just like this. Kevin, thank you. And then if you're on the East Coast, you might want to ask the New York Stem Cell Foundation, because they are similarly concerned with these clinics that are acting outside regulatory boundaries.
Yeah, I think Steve mentioned the ISCCR has a really good patient resource for this, and I think we've thought about going the other way instead of, because of the litigious nature of, marking ones that are bad, looking for ones that are good, and actually saying, can we do a good housekeeping type stamp on legitimate, resources. The difficulty is, to the point made earlier, there aren't many yet.
There aren't any, that's right.
So, I think figuring out how to navigate this area, I think information is the tool that we can give patient groups, and we work closely with a lot of foundations in that regard.
The ISSCR have a website called closerlook@stemcells.org, and that's a website designed for people with no medical training who just want to discriminate between legitimate treatments and fake scams.
I think the good thing about CIRM is that you'll get a personal response, and I think that makes a huge difference. Certainly when people contact me, I always respond. Sometimes I respond by saying, please ask Kevin McCormick at CIRM. But it makes a huge difference. They told me this, that it makes a huge difference that I paid attention to them and that I pointed them in a direction. And I think the problem with ISSCR, it's a wonderful organization, but you won't get a personal response from them. You'll just get suggestions. Yes, we're done. Thank you.